Hi, y'all. My name is Logan CJ, and this summer I was in the gun lab working with my mentor, Dan. And today I'll be telling you a cautionary symbio tale, the challenge of assembling better rubiscos. And so, what is photosynthesis and who is rubisco? To create energy, plants must undergo a process called photosynthesis, whereby the enzyme rubisco fixes carbon dioxide to RUBP in the Calvin cycle. And although rubisco is such a crucial enzyme in this process, it is wildly bad at its job, whereby it is slow, bad at distinguishing CO2 from O2, and is easily blocked. Furthermore, within these limitations themselves, there are also trade-offs between the speed and the ability to distinguish CO2 from O2. And so, how do you make rubisco? And then so to do this, we can utilize the rubisco biogenesis pathway, whereby 8 rubisco large subunits, RBCL, and 8 rubisco small subunits, RBCS, come together to form the rubisco L8S8 holoenzyme complex. And so specifically, the RBCL and RBCS formation is aided by chaperones or helpers, and one in particular is my friend RBCX. And so to help the process of photosynthesis, we can explore rubisco efficiency through this biogenesis pathway while targeting the chaperone RBCX. And so as mentioned before, rubisco has 2 major character flaws. One, its slow speed, and being bad at distinguishing CO2 from O2. And so while researching these trends, previous lab members had found that hornwort, pictured right here, normally has low specificity and medium speed. And you can see that it's different between Arabidopsis thaliana, which has the specificity, a higher specificity, but a lower speed. He had found that when you remove the helpers of rubisco formation, RBCX, you actually get increased speed without sacrificing its specificity. And this is very exciting because if we can take the hornet rubiscos that have faster speed without sacrificing the specificity and put them to crop plants, we can ultimately increase photosynthesis and crop growth. And this is very exciting to research. And so my mentor, Dan, is use crop plants such as tobacco as a proxy to do this. However, she does this with plant transformation, and it is rather slow because you have to start with transforming the plasmid, growing the plasmid all the way up to mature plants just to get to the end step to see if the hornet rubiscos are actually better, if the kinetics of rubisco are better. And so this process takes about 9 months, which is kind of rather long. And so we want to know, well, is there a way to speed up this, or is there a system that can speed up this? And so yes, there is. Going back to the rubisco biogenesis pathway, we can use a synthetic biology, a symbio system, to help this, whereby we take parts of the rubisco biogenesis pathway, RBCS and RBCL, along with the chaperones, put them into plasmids and then into an E. coli symbio system. And then this takes only about, allegedly, 2 weeks. So for my project, what we are going to do is take the hornet rubisco, put the tobacco chaperones, again with different combinations. So here we have our no X chaperones, and then with our X1 plasmid with chaperones and just RBCX1, and then same with X2 and X1, X2. So we're going to take the hornet rubisco, the tobacco chaperones, and then put them into our E. coli symbio system. And so our ultimate question is, will hornet rubisco have increased kinetics when assembled with the tobacco RBCXs / the helper of rubisco formation? And then so for our method, how did we do this? We took those combinations of our plasmids into our E. coli, grew them up, cell lysis, protein purification, and then our rubisco activity assays. And then so first, we're going to start with our plasmids and are growing them up. And then so to do this, I first needed to see if the plasmids had the correct sequence. And to do that, I did a mini prep protocol. And then I also used a software called SnapGene to see if the sequences were aligned. And then so as I said, this allegedly takes 2 weeks. However, this took the longest portion of my project because we had some bad luck while choosing colonies. But once we finally did have the correct ones, I then transformed and grew up my E. coli for protein expression. And so next, for our protein purification, we'd see this first visualization, and this is of preliminary data that shows that hornet rubisco can indeed be assembled with the tobacco chaperones themselves. And so this system was first done with RBCX1, And then so the visualization of our protein purification, we see our protein fractions here. First, we see our L8S8. So this is our Rubisco here. We can see that's visible. And then we also see HMW, higher molecular weights. And this consists of all the proteins that we don't want to really be with our Rubisco. It's separated. And then a different visualization of this is with an SDS page where we see the Rubisco large subunit and the Rubisco small subunit. And then so just as a reminder of why we're doing this in the first place, we want to see if the data from a Hornet Rubisco without the RBCXs have the increased speed without sacrificing the specificity of CO2. And then so moving forward with our plasmid combinations, we have our X1, X2 seen right here. And then we see the higher molecular weights, but we do not see the L8S8. We do not see our Rubisco. But however, with our no X plasmid right here, we see the higher molecular weights, and we see just a faint little something there for our L8S8. And then again, with our other two plasmids, our X1, higher molecular weights again, and then for X2, we also see the L8S8. And then so just as a reminder, we have our no, Rubisco, Rubisco, no Rubisco, Rubisco. And then so for moving forward, we only stored our no X and our X2 proteins for our spec assay. And then so you might be wondering, well, why didn't X1 actually work? And then so when we had gone back to the very beginning, even though the X1 sequence was aligned, when we put it into our E. coli biosystem, something had happened, and that was mutation. Here we see that there's actually a missing chunk out of our X1 sequence. So that was rather unfortunate, but we still moved on. And then so next we did our Rubisco activity assay. And then so here we first see our Rubisco turnover rate. And then another graph here shows our RUBP consumed against time, so the rate at which Rubisco fixes carbon dioxide to RUBP itself. And then so from here, we wanted to see, well, is our L8S8, is our Rubisco actually in our plasmids? And then so going back to our native page, so this is just the visualization of the purified protein. Here we see a marker and then a Arabidopsis rubisco that we're going to use as a reference. And then we see our preliminary X1 that shows that, yes, we can actually assemble Hornwort rubisco with tobacco chaperones. But then we also see here our plasmids that we move forward with. We're like, okay, is this L8S8? Is this the rubisco itself? And then so we kind of see some questionable, like a line here and maybe a smudge below. So we wanted to confirm, well, is our L8S8 there? And so to do that, we did a Western blot whereby we put antibodies that targeted the RBCL, Rubisco large subunit, and the Rubisco small subunit. And then so from here, you see in these red circles, we again have our Arabidopsis rubisco with our preliminary exon rubisco. And ideally, you should see on this similar plane, the line right here, saying that, okay, that is rubisco, but you actually see it above. And then the same with the RBCS. And then so from this, we see that even though Rubisco large subunit and Rubisco small subunit are present, they don't necessarily come together to form the complete L8S8 hollow enzyme of Rubisco. And then so just as a visualization, so actually no Rubisco, so L8S8 is not formed. And with that, I'll bring in the summary that E. coli sim biosystems have flaws. We see this through the missing plasmid of X1, the sequence isn't always there. And then also our sim biosystem yield is lower and does not function as expected. This could be because of the plasmid design, the chaperone sequence, or even the growth conditions themselves. And then so for some future directions, we can resequence our X1 plasmid and then repeat protein purification for higher yields. If we have higher yields, we can ultimately just do more experiments themselves. And then we can also explore the variabilities in E. coli sim biosystems and how that affects Rubisco efficiency. And then we can also conduct more informed research using tobacco transgenic systems. So if we can figure out how the tobacco RB6 chaperones have effect over the Rubisco formation, we can ultimately lessen the time that it takes to research and to increase that timeline. And then also ultimately at the end of the day, although it might seem like my summer research didn't have the most positive yields, but really it was far from a failure because I still did get to research a new symbiotic system and get just one small step closer to making better Rubiscos. And with that, I'd like to have my acknowledgements. The Gun Lab, of course, Laura, Dan, my mentor, Bryce, always, Sarah, Maddie, Zin, everyone at BTI, Delaney, Megan, Sylvia, of course, BTI and Cornell for hosting, NSF and USDA for funding, my home institution, UW-Madison as well. And then my last thing is everyone please get out your phones and follow my lab Instagram. I'm on the road to 20 followers. I see more than 20. Everyone please put your phones up. LinkedIn always, of course, and then the references. And with that, I want to thank y'all.
So questions for Logan?
A part of the process, you accelerated the 9 months to 2 weeks. If you tell me there's no for the 9 months, you would have had results.
I would say it's hard to say necessarily. I know it would certainly be rather unfortunate to get to the end of the 9-month process to see that your exome plasma didn't actually work, to see that you actually cannot confirm the results in the beginning, but also this was in E. coli, and then that would be in tobacco itself, so then that could also have different results. But yeah.
Great. I know this wasn't necessarily what your project focused on, but do you know if like your mentor tried other transformation methods as a means of shortening the amount of time required?
I know that a previous lab member had kind of set the foundation for the SynBio system, and we're actually comparing different SynBio systems. So in a way, yes, but then.
I have a question from a non-scientific mind. I'm curious, as scientists and people who do research, when something doesn't yield the results you want, like how does that feel? And like, how do you deal with that?
I suppose 2 answers. So one, as I guess as an REU student, obviously we only have 10 weeks here, so a lot of it is new. So then it's not only it's not really necessarily the results that matter the most. It's kind of it's a research experience for undergraduates. So it's really that experience of how can I work in the lab for like 40 hours a week, and how can I navigate that process? But then maybe for my mentor, can it be frustrating? Absolutely, because this is your PhD and this is what you're working to. But then it also it's like you can bounce ideas off your lab member. And so you can, okay, so this project or this system didn't necessarily work all the way, but you can still, okay, so this didn't work, but then it also showed you things, also like so many other things as well.